Hello students, today we will be looking into the nutritional significance of fatty acids and the digestion and absorption of fats. Lipids are large group of organic compounds predominantly made up of carbon, hydrogen and oxygen, and some contain phosphorus too. Lipids include a different categories of components like fats and oils, phospholipids, lipoproteins, waxes, sterols, glycolipids and sphingolipids. The term fat is generally used to denote that which is solid at room temperature, that is butter, ghee, margarine and lard. Oils are liquids at room temperature, the vegetable oils that we consume. Fats play a major role in the body. They are a source of energy, a concentrated source of energy. 1 gram of fat gives us 9 calories as against 4 calories that carbohydrates and proteins give us. But they are a storage form of energy. As steroids or steroid hormones, they play an important role in the metabolism. As phospholipids, they are part of the cell membranes. They also regulate the transport of numerous compounds across the cell membrane. Fat being a poor conductor of heat, it prevents the heat loss from the body. The layer of lipid below the skin helps in protecting the inner organs by offering structural support too. As component of myelin sheath, they provide insulation to nerve fibers. Fats aid in the absorption of fat-soluble vitamins, that is A, D, E and K. They are a source of essential fatty acids. They are components of lipoproteins, and HDL has a significant role in cardiovascular diseases. As eicosanoids, they regulate body temperature, aggregation of blood platelets and tissue inflammation. In food, fats improve the taste, flavor and color of the food that we consume. They give texture to the foods. They serve a medium to transfer heat while we are cooking food. They also increase the shelf life of the foods. Now, fats are made up of fatty acids, and that is why we say fatty acids are the basic building blocks of most of the lipids. Fatty acid is a carboxylic acid with a long aliphatic tail, which can be either saturated or unsaturated. Most naturally occurring fatty acids have a chain with even number of carbon atoms and normally ranging from 12 to 28 carbons. Fatty acids are usually derived from triglycerides or phospholipids. When they are not attached to either of the molecules, they are simply referred to as free fatty acids. These fatty acids differ structurally, chemically and functionally. They can be categorized in different groups based on various factors like chain length, degree of saturation, whether they are cis or trans fatty acids, and the configuration of the double bond. Depending upon the chain length, these fatty acids are classified as long, medium and short chain fatty acids. Short chain fatty acids are fatty acids with the aliphatic tail of fewer than 6 carbons. One of the examples is butyric acid, which is present in butter. Medium chain fatty acids are fatty acids with the aliphatic tail of 6 to 12 carbons, and these are called medium chain triglycerides. Coconut oil is a very rich source of medium chain fatty acids. Long chain fatty acids are the fatty acids with aliphatic tail of 13 to 21 carbons. Very long chain fatty acids are with the aliphatic tails longer than 22 carbons. Fatty acids can be either saturated or unsaturated fatty acids. Saturated fatty acids have no double bonds and they are saturated with hydrogen. Certain vegetable products have high saturated fat content like the coconut oil and the palm kernel oil. Animal fat products such as cream, cheese, butter, whole milk, dairy products and fatty meats are also a source of saturated fatty acids. Unsaturated fatty acids have one or more double bonds between the carbon atoms. Monounsaturated fatty acids, MUFAs, have one double bond in the fatty acid chain, with all the other remainder carbon atoms being single bonded. Polyunsaturated fatty acids, the PUFAs, have more than one carbon to carbon double bond. Polyunsaturated fats can be seen in nuts, seeds and fish. This is just an example to show you how saturated and unsaturated fatty acids differ. If you can see the double bond between two carbons, and in saturated there are no double bonds at all. Another category is the cis and trans fatty acids. A cis configuration means that the two hydrogen atoms adjacent to the double bond stick out on the same side of the chain. A trans configuration, by contrast, means that the adjacent two hydrogen atoms are on the opposite side of the chain. Most fatty acids in the trans configuration are not found in the nature and are the result of processing. They raise cholesterol and they also lower HDL. Thus, trans fats are a matter of concern because they increase the risk of cardiovascular diseases. The figure just gives an example about how the hydrogens are positioned with compare to carbon and the double bond. Then essential fatty acids. These are the fatty acids which are required for the body, but at the same time they cannot be synthesized by the humans and they have to be obtained from food, and that is why these are called as essential fatty acids. One category is the omega-3 fatty acids. They are also called as N3 fatty acids. These are the PUFAs with a double bond, C double bond C, at the third carbon atom from the end of the carbon chain. The three types of omega-3 fatty acids which are involved in human physiology are alpha-linolenic acid, icosapentaenoic acid and docosahexaenoic acid. Both are found in marine oils. Omega-6 fatty acids, also referred to as N6 fatty acids, are a family of pro-inflammatory as well as anti-inflammatory PUFAs that have a final carbon to carbon double bond at the N6 position, that is the sixth bond counting from the methyl end of the fatty acid. The biological effects of the omega-6 fatty acids are largely produced during and also after the physical activity for the purpose of promoting growth during the inflammatory cascade to halt cell damage. They also promote cell repair by their conversion to omega-6 icosanoid that binds to the diverse receptors found in every tissue of the body. So if we just briefly look into how the fats and fatty acids are classified, we have saturated fats like animal fats, butter and lard, and you have unsaturated fats. Unsaturated can again be divided as mono and polyunsaturated fats. Polyunsaturated again can be omega-3 fatty acids that are very rich in fish, shellfish, flax seeds, soya beans, walnuts. Omega-6 fatty acids, safflower oil, sunflower oil are rich in omega-6 fatty acids, and monosaturated are your omega-9 fatty acids which are present in peanuts, olive oil and almonds. Now if we just look at the functions of omega-3 and omega-6 fatty acids, because they are going to play a major role in human physiology, let us look at omega-3 fatty acids. These are the alpha-linolenic acids. They are anti-inflammatory, they promote blood thinning, they cause artery dilation, they lower blood pressure, they improve arthritis pain, and they elevate depression and stabilize the moods. As against this, omega-6 fatty acids, that is the linoleic and the arachidonic acid, these are pro-inflammatory. They promote blood clotting, artery constriction, higher blood pressure, increased joint pain, and also cause increased depression and mood swings. Now let us come to the next component of the lesson, that is digestion of fat. Before that, let us briefly look into what digestion means. Digestion is the breakdown of large insoluble food molecules into small water-soluble food molecules so that they can be easily absorbed into the blood plasma and distributed all over the body. It is a form of catabolism, which is often divided into two processes based on how the food is broken down. That is, there is a mechanical digestion, which refers to the physical breakdown of the large pieces of food into smaller pieces, which can be subsequently assessed by the digestive enzymes. And the chemical digestion, where the enzymes break down food into smaller, easily absorbable particles. There are different phases of digestion. The first one is the cephalic phase, which occurs at the sight, thought and smell of food, and it stimulates the cerebral cortex. Taste and smell stimuli are sent to the hypothalamus, medulla oblongata, and then it is routed through the vagus nerve. The actual phases are the gastric and the intestinal phases. So the digestion begins in the mouth, where the secretion of saliva and the digestive enzymes is seen. Food is formed into a bolus by the mechanical mastication, and it is swallowed into the esophagus, from where it enters the stomach through the action of peristalsis. Gastric juice contain HCl and pepsin. These would normally damage the walls of the stomach, but the mucus that is secreted protects the mucosal lining. In the stomach, further release of enzymes break down the food, and this is combined with the churning action of the stomach. There is a partial digestion of the food that takes place before it enters the duodenum as a thick chyme. In the small intestine, the larger part of digestion takes place, and this is helped by the secretion of bile, pancreatic and intestinal juices. The intestinal walls are lined with villi, and their epithelial cell cover numerous microvilli to improve the absorption of the nutrients because they increase the surface area of the intestines. In the large intestine, the passage of food slows down to enable fermentation by the gut flora to take place. Here, water is absorbed and the undigested waste material is stored as feces to be excreted. Now let us look at digestion of the fat specifically. Most of the fat in the diet is in the form of triglycerides, and there is small amount of cholesterol and phospholipids. In the mouth, digestion of some amount of fat can happen because of the lingual lipase, which breaks down the small short chain lipids into diglycerides. However, this is not of significance in adults because in small children, the lingual lipase digests the short chains and the medium chain fatty acids, which are present in the mother's milk. But the digestion of fat as far as adults are concerned occurs in three phases: the gastric phase, the duodenal phase and the ileal phase. In the stomach, the gastric phase, there is emulsification of fat that occurs as the chyme is formed because of the churning of the stomach contents. When the food enters stomach, the gastrin hormone is released, and this releases the human gastric lipase. It hydrolyzes just about 10% of the fat that is consumed. It primarily acts on short chain fatty acids. In the small intestine, the maximum digestion of fat occurs. The presence of fat in the small intestine produces hormones, most important being the cholecystokinin. This stimulates the release of pancreatic lipase from the pancreas. It also stimulates the release of bile from the liver because it helps in the emulsification of fats and also for the absorption of fatty acids. Complete digestion of one molecule of fat, that is a triglyceride, results in 3 fatty acid molecules and 1 glycerol molecule. Bile is a greenish yellow thick sticky fluid. It is consisting of bile salts, electrolytes, bile pigments, cholesterol and other fats. The gallbladder stores bile. Bile flows out of the liver through the right and left hepatic ducts, which come together to form the common hepatic duct. This duct then joins with a duct coming from the gallbladder, which is called the cystic duct, and this forms the common bile duct. The pancreatic duct joins the common bile duct, whereby it just empties into the duodenum. Between meals, bile salts are stored in the gallbladder and only a small amount of bile flows into the intestine. Food that enters the duodenum triggers a series of hormonal and nerve signals that cause the gallbladder to contract. As a result, bile flows into the duodenum and mixes with the food contents. Bile has two important functions. It assists in the digestion and absorption of fat, and it is responsible for the elimination of certain waste products from the body, particularly hemoglobin from the destroyed RBCs and also the excess cholesterol. Bile also has other functions. Bile salts make cholesterol, fats, and fat-soluble vitamins more soluble or more dissolved and thus help in absorption. Bile salts stimulate the secretion of water by the large intestine to help move the contents along. Bilirubin, which is the main pigment of bile, is excreted in bile as a waste product of destroyed RBCs, and this is what gives the stool a green-brown color. Drugs and other waste products are excreted in the bile and later eliminated from the body. Various proteins that play important role in the bile's absorptive function are secreted in the bile itself. Bile salts are reabsorbed by the last portion of the small intestine, extracted by the liver, and again resecreted into bile. This recirculation of bile salts is known as the enterohepatic circulation. All the bile salts in the body circulate about 10 to 12 times a day. During each pass, small amounts of bile salts reach the large intestines where some are reabsorbed and the rest are excreted in the stool. It is important for us to know the role of bile because bile is the one which is going to play a major role in the emulsification of fats and thereby promoting digestion of fats. Lipids are basically hydrophobic, that is, they are poorly soluble in aqueous environment of the digestive tract. The digestive enzyme lipase is also water soluble and can only work at the surface of fat globules, and that is the reason why the fat has to be emulsified. And this digestion is greatly aided by the emulsification, that is, the breaking up of fat globules into smaller droplets, and this job is predominantly done by the bile salts and phospholipids. Bile is amphipathic or what is known as amphiphilic molecule. It is water soluble. So the motility in the small intestine breaks the fat globules apart into very small droplets, and they are coated with bile salts and the phospholipids. They prevent the emulsion of the droplets from reassociating, and at the same time, they being water soluble, they allow the lipase to act on these small droplets of fat, and that is why we consider bile as a very important factor in the digestion of fat. So emulsification greatly increases the surface area where the water soluble lipase can work, and it can digest on the triacylglycerides or the triglycerides. It releases monoglyceride and two free fatty acids. These fatty acids are released from the first and the third carbon of the glycerol. The fatty acid attached to the second carbon remains intact. Another factor that helps is colipase, which binds and anchors lipase at the surface of the emulsion droplet. All this support system is required so that the fat is properly digested and absorbed. Dietary phospholipids are hydrolyzed by the pancreatic phospholipase 2. Cholesterol esters are digested by the pancreatic cholesterol ester hydrolase. This is the structure of a triglyceride. We can see the glycerol molecule to which three fatty acid chains are attached. So when the lipase acts on these triglyceride, the free fatty acids, that is at first carbon and the third carbon, are released, and what is left is the fatty acid at the second carbon, and that is why we call it as a 2-monoglyceride. So when lipases act on a triglyceride, we get two free fatty acids and 2-monoglyceride. The number 2 indicates that the fatty acid is attached to the second carbon of the glycerol molecule. Short chain fatty acids and medium chain fatty acids directly enter the portal circulation. Long chain fatty acids, that is, fatty acids with a chain length of more than 12 carbons, cannot enter the enterocytes directly. They associate with bile salts, micelles are formed, and then they are absorbed into the enterocytes. The micelle may also contain cholesterol and fat-soluble vitamins. The monoglyceride and the free fatty acids enter the enterocyte in the small intestine. There, reesterification of the 2 fatty acids and the 2 monoglyceride occurs in the enterocyte itself. This is catalyzed by the enzyme acyl-CoA cholesterol acyl transferase. Unsaturated fatty acids are digested better than the saturated fatty acids. Again, one small important note: short chain and medium chain fatty acids, because they do not require the presence of bile, are particularly given during the treatment of liver diseases. Bile is secreted by liver, and in liver diseases, secretion of bile can be affected. But at the same time, fat requirement can be taken care of by incorporating medium chain fatty acids in the diet, because they directly enter portal circulation. Phospholipids are similarly reesterified in the enterocyte. 90% of the monoglycerides and the fatty acids are reesterified. The triacylglycerols, phospholipids, cholesterol are assembled with a protein to form chylomicrons, which is a phospholipid. Chylomicrons enter the circulation and are distributed to all parts of the body, and that is how they distribute the fatty acids to the body. Lipoprotein lipase present on the surface of the capillaries act on the lipids in the chylomicrons and releases free fatty acids. These are very effectively utilized by the muscles and other tissues. The digestion and absorption of fat is predominantly influenced by the amount of fat consumed. More fat, longer is the time taken for digestion. Also, most of the fat then may not be digested. Then the kind of fatty acids. Long chain fatty acids take a much longer time for digestion. As we had seen earlier, short chain and medium chain fatty acids are very easily absorbed into the portal circulation. Similarly, unsaturated fatty acids take a longer time to digest. If associated with high calcium intake, calcium can bind with fatty acids, they form soaps, and they thus hinder digestion. The presence of emulsifying agents like bile, they aid in the digestion. If there is any problem with either the liver or the gallbladder, then the digestion of the fat can be affected. This is just a diagrammatic representation to tell how the fats are digested and absorbed. Lingual lipase, we talk about important in children but does not play a major role in adults, because lingual lipase digests short and medium chain fatty acids which are predominantly present in the mother's milk. The role of bile and cholecystokinin, gastrin stimulates the gastric release, then the liver produces bile, sends to gallbladder because of the response to secretin, and there is digestion by the pancreatic lipases. So in summary, we can see that lipids are organic compounds which include fats and oils, phospholipids, sphingolipids and waxes. Fats have multiple functions at the cellular level, and they also improve the taste and flavor of the food that we consume. Fatty acids can be classified depending upon the length of the chain, degree of saturation, whether they are cis or trans, and whether essential or non-essential. Fats can be digested in the mouth by children. Most of the digestion, however, takes place in the intestines. Bile helps in the emulsification of fats, and this aids the pancreatic lipase to act on fat. Free fatty acids and 2 monoglycerides are absorbed into the enterocyte, where they get reesterified again. Quantity and the type of fat, presence of emulsifying agents have a great influence on fat digestion. Thank you.